All right. Perfect. So it's now recording. And so hi, everyone. Good evening. Thanks a lot to all of you for attending this month's NeuroMeetup talk. So the title of this session is Electroceutical Strategies to Optimize Deep Brain Stimulation for Movement Disorders. So the first speaker is going to be Fernando Rodriguez from the University of Oxford. So Fernando has a background in Mechanical and Biomedical Engineering from the ETH in Zurich. He first encountered movement disorder electrophysiology and deep brain stimulation while he was a research assistant in Zurich and at the University Hospital of Zurich, which led him to completing his master's thesis under the supervision of Professor Peter Brown at the University of Oxford. He's now doing his PhD in the lab of Professor Huiling Tan, still at the University of Oxford, where he's working on signal processing and machine learning for real-time decoding of neural signals in the context of adaptive deep brain stimulation. And so, Fernando, the floor is yours.
Thanks, Camille. So over the next 20 minutes, I'm going to attempt, perhaps somewhat clumsily, to explain what DBS is, sort of what its main clinical application is like, where it comes from, so a bit of the history, and then how DBS enables us to think about movement disorders through the lens of neurophysiology, as opposed to other lenses that one might take in neurosciences. And then I'll go a bit into the concrete research I do, which deals with signal processing for adaptive deep brain stimulation. Excellent. So briefly, to give some background, although I am assuming a bit of familiarity with deep brain stimulation, if not, feel free to stop me. It is a surgical procedure that is used usually to complement pharmacological treatments. This is very much the case for Parkinson's disease, a little bit less the case for essential tremor or dystonia, but is basically standard of care for these disorders in order to try to manage the motor symptoms that come along with the disease. Now, it has been proven to work quite well under a number of over the years. One would say this treatment has over 20 years of history, and it is currently being expanded to another set of both motor and psychiatric disorders, including Tourette's, Huntington's, MDD, OCD, addiction, pain, schizophrenia, etc. So it's really a broad sort of people are trying to port the success that it has had in the narrow movement disorders fields to other domains of clinical neurosciences. So a bit about the history. DBS has its roots in ablative surgical procedures, mostly polydotomies and thalamotomies, which is a procedure whereby first with, crude neurosurgical instruments, one would go into a patient's brain and try to take out a cluster of cells that is suspected to be generating or to be relaying aberrant activity, with the ultimate goal of alleviating motor or otherwise other types of symptoms. Now, in 1987, the story goes that Louis Benabit in Grenoble in France serendipitously discovered DBS. What that basically means is that at that time they weren't using these crude neurosurgical tools anymore. They were using electrodes, and they would send electrical impulses, low frequency but high amplitude ones, in order to find the proper place. And once they were at the proper place, which is called intraoperative target mapping, they would send a big pulse in order to burn some cells and ablate that area. Now, Benabit decided to play around a little bit while in theater, and he started playing around with different frequency ranges, and he found that high frequency stimulation, approximately at the time it meant 100 hertz, although today the therapeutic stimulation is generally believed to be around 130 hertz. He found that that would very much mimic the effect of an ablation, with certain advantages. First of all, it would be adjustable. and it would be reversible. Once you get rid of the cells, you can no longer get them back. If you turn the stimulation on, you can, however, still turn it off again. Now, the part where this becomes interesting for me as an engineer is when they start sticking all of these electrodes into people's brains, and either in theater or a few days after surgery, where the cables are what we would call, where the patients are what we would call externalized, meaning that we have access to the cables looking out of the burr holes on the skull, and we can attach ourselves to them and look at the electrical activity patterns that are perhaps associated with pathology, that are perhaps associated with function, all of that. And this is what sort of leads into this neurophysiology or electroceutic centric view of disease, where you no longer are looking at Parkinson's, let's say, in terms of behavioral criteria, but you're measuring it in terms of the proportion or preponderance of specific oscillatory activity that you have determined to be bad. So a bit more succinctly put, or at least better put than I ever could, this is from a psychiatrist speaking in the late 70s, who says, after implantation of chronic electrodes, long-term depth recordings and repeated electrical stimulations enable the psychosurgeon to accumulate knowledge about the pathophysiology of the brain and to improve the treatment of the patient in question. It may even be possible to treat the patient with repeated electrical stimulation without macroscopic destruction of brain tissue. So that's the whole of 30-second pitch of this neurophysiology-centric view and DBS. Now, going into this a bit more concretely, the story goes as follows. People have access suddenly to these brain signals. They start looking at them, and the very first thing that jumps out to them is that there's an association between dopamine levels within the basal ganglia and oscillatory activity, specifically in one band, that would be beta, between 13 and 30 hertz, but also in gamma around 70 hertz. It was first reported. This is work done by my former supervisor, Peter Brown, in the early 2000s. And then it was also found that there's a relationship between symptoms, especially in the case of PD, bradykinesia, rigidity, and the same oscillatory activity pattern, leading to the view that beta activity has a putative role as a sort of antikinetic oscillation and gamma as a prokinetic oscillations. So what becomes clear at this point is that there is, at least correlationally, a relationship between oscillatory activity, symptoms, and sort of a more classic view of disease in the brain, which relates to neurotransmitters, etc. And this is where we change a bit into the neurophysiology-based paradigm that I'm going to get into. So currently, the way that we do research in this field is we either intraoperatively in externalized patients, or over the past few years in patients who have a new generation of devices that enables streaming capabilities of data with some limitation, but still there it is. And we look at the electrical activity in the brain, we look at the patterns, we try to find correlation between clinical behavioral covariates, pathology, oscillations, all of that. And this approach has taken us quite far. So here I have a sort of mandatory video of an essential tremor patient who is very much struggling to sign because of his tremors. And then you'll see shortly that he takes a remote control, puts it to his chest, which is where he has an implantable pulse generator or IPG, turns the stimulation on, and thereby the tremors quite quickly disappear, and he's able to sign his name. So this can work at times. Yeah, there we go. There are, however, some issues with this concept of having an IPG constantly delivering high frequency pulses into your brain. Intuitively, one might simply think that if you're delivering current into the brain and you're thereby sort of overwriting or interfering with natural brain oscillations, you might kill some of the bad ones, but you might also kill some of the good ones. And this is one of the things that we want to avoid. We want to minimally disrupt functional activity and maximally disrupt pathological activity, whatever that might be. In clinical terms, this translates to a number of impairments for patients that vary. Oftentimes they relate to speech, gait, postural instability, or in terms of non-motor symptoms, you might have impulse control issues, apathy, cognitive issues, and stuff like that. There's another effect called habituation, for which we oftentimes blame the fact that you have current being delivered into your brain for many years at a time at the same settings. And this creates sort of long-term plastic effects that diminish the therapeutic effect of the stimulation. And then there's also this new idea that for psychiatric diseases which are a bit more complicated, where we don't have this very clear beta oscillation that is our enemy and that we want to suppress, it might be better to, or it might be necessary in order for this approach to work, to come up with stimulation strategies that are a bit more fine-grained, that are really quite selective in terms of the patterns of activity in the brain that they try to stimulate. So broadly, these are the challenges, and there's two main approaches that the field is taking towards solving them. The first relates to where to stimulate, and the other when to stimulate. Now, I'm not going to say a whole lot about the where to stimulate because I don't know much about it, to be honest. It relates to sort of better mapping of the circuits that are associated with even individual symptoms and how they vary for individual patients. And it's a lot of imaging work, although also a little bit of electrophysiology work. Alberto is going to talk about that. But the part that I'm interested in is the when to stimulate part. So when to stimulate. Here we have the word to stimulate again. We might see some of the different functions that the basal ganglia are involved in, how they might get disrupted through disease, and how perhaps, given a very particular or a very fine-grained knowledge of what the stimulation is and where to deliver it and how to deliver it, one might be able to very selectively modulate all of these symptoms to get to a sort of optimal quality of life place. Now, the when to stimulate part interests me more because I come from a signal processing background, and this idea relates to, instead of delivering stimulation chronically, to really try to ascertain what components of the signals we want to respond to, and when we encounter them in real time, to deliver stimulation optimally so as to suppress them. So that's the whole idea behind adaptive or closed loop deep brain stimulation. The closed loop part comes because we are no longer constantly stimulating, but we are stimulating according to some decision that we make based on signals, control signals that we extract from the intrinsic brain dynamics, although we might also extract them from other types of sensors, such as peripherals and so on. Now, briefly, a bit of a mute point, although I think it has some historical interest, which is DBS wasn't thought of in the very, very days as a chronic sort of treatment approach. So in ethically dubious experiments in the 1950s and 60s, psychiatrists such as Robert Heath at Tulane University or Jose Delgado at Yale University in the US, they would come up with these contrived experiments where they would give either patients the ability to deliver stimulation pulses to their own brain or to other people's brains or animals. Here we have an example where monkeys are given the ability to sort of turn off supposedly the aggressive behavior in other monkeys. And Jose Delgado's big point about that was that this potentially could be used to, create a more psycho-civilized society. I would call it a better behaved society of sorts. The point for us being is that this stimulation from the very beginning, or the very idea of actuating on the brain, has always been considered a thing that you should do at specific times because the brain is complicated and you just don't want to be sending out energy willy-nilly. So this for my sort of historical perspective. But now getting back to ADBS. So the goals of ADBS are broadly to increase the therapeutic window of the stimulation. This includes, on the one hand, to enhance efficacy by prolonging the duration of the effective stimulation, by selectively targeting pathological rhythms and suppressing those very effectively, by minimally interfering with functional activity and thereby reducing side effects, And also there are other points such as adapt to context. So for instance, one of the things that is being explored is throughout the night, should the stimulation be on, should it be on in the same way, etc. Now, in terms of the feedback strategies that we use, meaning the signals that we look for within the brain oscillations in order to guide the stimulation, it's either a direct marker of pathology, which would be this beta oscillation, for instance, that we've talked about before. But what I do rather is I look for proxies that are behavioral, that we can decode from the brain oscillations and that we know will be useful times to deliver the stimulation. So one classic case of this would be an essential tremor patient. We know when he moves, when he begins moving, it is likely that the tremor will set on quite quickly after that. So what we want to do is detect the periods when the patient starts moving and deliver the stimulation. Then here we have a schematic from a colleague of mine at the lab, Sheng Hong, where we basically see how such a system works. We get LFP data, we extract features from it, and then we have a few classifiers trained depending on whether the simulation is on or off, and depending on what the classifier is trying to detect. And this classifier gives us a control signal that we use to either switch the stimulation on or switch the stimulation off. So that's the basic idea behind adaptive deep brain stimulation, or movement triggered adaptive deep brain stimulation in this case. Now, thinking about this a bit more generally, what do you need? But you need a system that is able to, in real time, process incoming LFP signals, which are the type of signals that we get from depth electrodes, and extract from them some type of marker that can either be directly used to titrate stimulation or that gives us some indication about what the patient is doing, in which movement or behavioral state he's at, and we might use that information in turn to trigger or adjust the stimulation. A few constraints on this are, well, first of all, we might want to include other information in this, but these systems should be personalized, meaning each patient should have a system that works for him or her. It's very difficult to train systems that work across patients, just because there are great divergences with respect to where the electrodes are placed and what activity patterns we see across patients. The patterns should directly be inferred from the signals, meaning if we have to predetermine what they are, this puts us sort of in a tricky spot, because we need to because you become reductive in the sense, if you say I only want to look at beta, you become on the one hand precise in terms of only targeting beta, but the point is the signals are complicated and there might be predictive power within them beyond beta. That's the idea. And then sort of in terms of an engineering puzzle, this is an interesting problem because you have issues like real-time compatibility, which includes sort of computation and all of this, but I'm not going to go into those in detail. So the current approach for doing this, I've already briefly mentioned this, but we have a filter bank, which is what I call it, which extracts features from waveforms that are incoming in real time, thus generating a feature vector. This gets fed into a classifier that has previously seen this feature vector movement state pairs in our case, and hopefully at inference time is able to tell us something about what the patient is currently doing, and we use that information to adjust the simulation. There are, however, some questions about the optimality of the filter bank, and this is sort of the whole thesis of my PhD. How much information is contained within these oscillations, and what are optimal ways of extracting it? Optimal both in terms of computation, but mostly optimal in terms of doing a good job of extracting information. Now, my little bit of contribution is really quite simple, is instead of using predetermined filter bank, we simply parameterize it, and then we apply an optimization algorithm in order to find the optimal parameters that so as to basically increase the performance, because we're not making random assumptions about how to look at the waveforms. We're letting a deep learning system, in this case, infer how to look at them. How this looks a bit more concretely is, well, this parameterized filter bank can be thought of as a cascade of FIR filters. That's in essence what convolutions are, with a few details here and there, nonlinearities and normalization layers mostly. And you can really think these filters as having a frequency response, this and a phase response the same any other filter does. And for a subset of the filters, we even constrain the shape of the filter element so as to give us certain properties in terms of frequency. It turns out that these types of filters are quite well suited to solve a number of tasks whenever you're looking at waveforms. For instance, if I were to give you a waveform like this, which begins very sinusoidally and slowly becomes very sharp, and then becomes very sinusoidal again, and I asked you, how could you quantify the degree of sharpness in this wave? Well, you could do it, and there are ways of doing it, but the whole sort of idea of this approach is you just train a number of filters that autonomously become quite good at extracting wherever correlate you are asking it to extract. So this is one such example. This would be simply a frequency selection task, because the difference between a sinusoidal wave and a sharp wave is basically in the harmonic content, but other types of effects might be captured. Here I aim to demonstrate a few waveforms that we generate, and where we show that this parameterized filter bank or convolutional neural network approach might be better than a feature-based support vector machine. So here we have some basic patterns that we oftentimes observe, which is again modulation around 21, 22 hertz, which is approximately what we might call high beta band activity, and then some modulation about 70 hertz, which might be called gamma. We generate two phases, add some noise to them. This is here the noise scale, and we see that the CNN ekes by performance out a little bit the support vector machine. This difference becomes a bit more apparent for other tasks. So the waveform sharpness I already mentioned, you can see the harmonic distortion, and then you can see that the ConvNet is a little bit better at extracting information. Same history here. How do we optimally extract phase amplitude coupling patterns from oscillations? Well, here I show that again, these convolutional filters are a bit better optimized than our previous approach. So on and so forth. So here we have cross-channel theta gamma phase amplitude coupling instead of intra-channel. Here we have nonlinear phase, where the peak to trough time difference within the waveforms is very different than the trough to peak. So basically a nonlinear phase. And here again, we see the harmonics, the CNN being a bit better at solving these sorts of issues. And this is sort of the stuff I'm interested in, which is to say how to optimally extract features or extract relevant oscillatory patterns from multi-channel time series data, or from the sort of data that we encounter within these deep brain LFPs. So in brief, these convolutional filters or these ConvNets are quite good at keeping track of oscillations, waveform characteristics, phase characteristics, or combination of these. We put this approach to the test with some real data, meaning 8 essential tremor patients whom we visited usually a day after surgery, and we have asked them to move versus be at rest, moving us in 3 different ways. And we've done this with the stimulator being on and with the stimulator being off. Which is relevant because you get a big stimulation artifact and you need to modify your pipeline so as to be able to deal with it. And then we are training classifiers. At the end of the day, we have training data that is approximately 6.7 minutes long, and then validation data that is approximately 1.7 minutes long. And if we look at how the two systems perform against one another, here on the x-axis, we have again the support vector machine scores, on the y-axis, the convolutional neural network scores, and we see that there seems to be a little bit of an upward trend. So there are a few cases when the ConvNet is being able to extract relevant information from the oscillations that are not readily accessible to the support vector machine because of the features that we have manually constructed. They don't seem to explain this part of the variability very well. This is sort of to show that this holds true across the cross-validation strategy, because of the short data length 5-fold cross-validation strategy. And yeah, so in terms of getting to an end of this presentation, I hope I have more or less managed to explain how access to these deep brain signals enables innovations such as adaptive deep brain stimulation, and shown some of my work that aims to improve the decoding so as to better drive these adaptive deep brain stimulation systems. And currently we're working on bringing all this into the clinic and working with real patients and all of that. Thank you very much.
Right. So thanks a lot, Fernando. That was really insightful. It's really cool to see that CNNs can be used to decode and to apply a DBS. So to people in the audience, please feel free to ask any question you might have, preferably in the chat, so that we can relay them to the speaker. After each talk, we are going to have a short, 10 minutes of question and answers. So we don't have any question in the chat for the moment. So I'm going to start with a very short practical question. Just wanted to have a broad idea of whether, first of all, DBS is a common procedure. How many persons live with DBS around the world? How often are such procedures realized in the world? And especially if patients already live with a DBS.
So with respect to the first question, Jesus, I knew the figure a week ago or so. Alberto, perhaps, I'm willing to say in the last 20 years, about 250,000 patients have been implanted worldwide. It's something like that. So it's not like 10 or 20 at this point.
Sounds reasonable, yeah.
And with respect to the DBS already used in clinical practice, there's a few trials ongoing, one based in Amsterdam, done by Medtronic. Well, it's a multi-center trial where they are trying to show, it's a non-inferiority trial of beta-triggered ADBS, meaning the stimulator only turns on when it detects an excess of activity in the beta band versus continuous stimulation. So it's not yet in the clinic, but it's on the long regulatory way to perhaps get there someday.
Okay, thanks. We have a question from Alberto, if you want to ask it directly.
Yeah, sorry, I was still writing something, but I was curious to see if you know, or if you approximately have measured what is the computational cost of your approach, and if you could maybe compare this to, let's say, conventionally used approach for adaptive DBS, maybe based on the detection of the beta rhythm. With respect to the last part of your question, that's very difficult because all of the software that they use in these embedded devices is proprietary. So it's very difficult to actually know what's going on in there and what sort of assumptions they're making, what sort of filters they're running, what sort of signals they get and how they process them. It's really a bit. I think it's very relevant, of course, being a signal processing person myself, but many people seem to just gloss over and say, if it gives you a beta estimate, it must be good. But we know that extracting beta estimates isn't always trivial. So that's with respect to the last part of your question, with respect to deploying these convnets in real time. So as to, and how much that costs. Well, here we have a sort of initial large network. This is the one that I use compared against the either bandpower driven approach, which is very inexpensive computationally. So you have the number of floating point operations that it takes you to go from the raw signals to your classifier output, and here is approximately 2 million, here 500 million, watch the log scale, and a reference approach from the literature where they use basically these computer vision systems. It's much higher. It's 20 million. However, we have been doing some work on bringing this number down, and I believe we've done so quite successfully. So by deploying model distillation techniques, which basically means taking a large model that has been optimally designed so as to train very well, but not so as to work at a low compute cost, and then training a small model that is optimized to work at a low compute cost, we can train the smaller model to try to imitate the large model. And it can do so quite well because it has access to the whole distribution that the large model has learned, not only to the raw binary labels that you would train the large model with. And applying this technique, we can see that we can bring the computational cost of the convnet down. The point at which we really start to take a hit is approximately 3.5 times of the computational cost of the support vector machine. So at this point, I wouldn't even call it a deep neural network. Deep learning has gone out the window. It's just, let's say, 3 FIR filters cascaded with nonlinearities in between. And it turns out that those are all you need in order to extract, to find most of the sources of variance that are relevant for your classification task.
Yeah, thank you.
Yeah, thanks for the questions. Good ones.
All right, thanks a lot. We don't have any more questions from the audience in the chat, so I suggest that Alberto start the next talk, if that's okay for you. So I'm going to briefly introduce Alberto, the next speaker. So Alberto completed his master's degree in neuroengineering at the University of Genova in Italy, and during his PhD at the Italian Institute of Technology, and then at the University of Kansas Medical Center, he had the opportunity to investigate how the electrical stimulation of neuronal tissue could promote recovery after brain injury, using both in vitro circuits as well as in vivo cortical networks. So these studies allowed him to move to the University Hospital of Bern in the lab of Gerd Tinkhauser to make a translational step from animal model to human research. And nowadays, as a postdoc, he's currently investigating the characteristics of field potential activity in the basal ganglia network of patients with Parkinson's. And the twofold objective of such project is aimed at characterizing the most entailed features of the neuronal signals in the disease, and at the same time to define novel potential sources of control signal for ADBS. Thanks a lot, Alberto, and the floor is yours.
And sorry for the very long bio.
It's okay.
I try to be very specific on my work, but then at the end it's easy to talk about other things. So by the way, so thank you for inviting me to this talk and to give me the opportunity to share my research. First of all, can you see my screen? I think so, if nobody has complained about that.
Yeah, it's perfect. We can go ahead.
Okay, so I'm kindly asking you to keep your question for the end of the talk, if possible, using that chat. And I would like to start my talk by showing you an example of what is the let's say the current stage of DBS for Parkinson's disease. Then I'm going to list some of the main criticality related to DBS programming in the clinics towards the sensing-based optimization for DBS. And then I will finally talk about my main project, which regards mainly the spectral somatotopy of the STN. So, since Fernando already gave you a very detailed overview on the DBS, I would like to start this talk just by showing you a practical example of what's the impact of DBS on the main symptoms of Parkinson's disease, just to convince you that this treatment might be somehow magic. And as you can see on the left, the DBS was off while we set on, we set the stimulation on the right, and the symptoms of the patient totally disappeared. So this is very impactful. So anyway, as it is true that we need to be careful with anything which is injected or implanted in our body, if we want to get the most by deep brain stimulation, it has to be delivered in a certain way. So it is known that stimulation delivered to the wrong brain tissue, for instance, might, let's say, induce very severe side effects. And for this reason, the technology of DBS electrodes and also the techniques is still improving. So novel directional leads, for example, are composed by 8 contacts and allow to shape the stimulation field, thus reducing the risk of potential side effects and increasing the spatial resolution for the stimulation. And if from one side one could benefit from such an improvement, this might also increase the complexity of the treatment. So usually, in order to achieve the optimal stimulation effect and the optimal therapeutic effect, the stimulation frequency, the amplitude, and the pulse width must be adjusted on each specific patient. And this is done by a movement disorder specialist. And to define the contact with the highest clinical efficacy, this person usually increases the stimulation until what is called the effect threshold. And then he increases more and more again to reach the side effect threshold. But then, as you can imagine, an electrode with an elevated number of contacts will require an increased, let's say, an increased time for testing the stimulation of all of the possible combinations that you can have. And so at the end of the day, it has been proven that clinicians prefer to use what is called ring mode testing modality, so stimulating at the level of the whole ring, thus without fully exploiting this technology. So for all these reasons, we can say that DBS may require tools to overcome and exploit the clinical benefit of the therapy. And there are many tools that potentially could be used for this purpose, like imaging, for example. But from the others, novel DBS approach, you use the electrophysiology. It is important to notice that DBS electrodes not only allow to deliver the stimulation, but also to record the brain activity from the subthalamus, from the target. And this activity is in the form of the so-called local field potentials, or LFPs. Very briefly, in few words, amplitude and frequency of this local field potential really depend on the contribution of multiple sources and from many properties of the brain tissue, starting from the fast action potential to the slowest fluctuation of the glia, for example. So why do we think that brain activity should be important for us? So, well, for example, to push for a more personalized treatment, which might be guided, for instance, by pathological activity, as mentioned before by Fernando. This could be useful for both DBS programming as well for the closed-loop DBS strategies. And for instance, for DBS programming, the main benefit might be the reduction in programming time, as well as the standardization of the procedure that allows the requirement of less specialized clinicians or experts. While for the closed-loop strategies, these offer the opportunity to optimize the DBS by automatically adjusting the timing and the parameters of the stimulation in real time. So this based on the biomarker, thus minimizing what is the side effects. So which are the most important electrophysiological features to be targeted? Okay, so beta band, as you already heard, is considered the most promising biomarker. And this is already implemented in the first trials of the adaptive stimulation protocols. So Parkinson's disease is known to be characterized by an exaggerated peak in the beta band. This rhythm is also reduced by the pharmacological treatment and by DBS, and it correlates with motor deficits and is also associated with the highest clinical efficacy of the stimulation. So, but despite these, if one think of sensing DBS using the beta band, there might be some limitations. So beta band might be inconsistent among patients, especially consider different phenotypes, and might not represent really well the clinical scores of the patients. And one should also consider the interaction between a lot of rhythms within the brain, which can interact with the detection of the beta band, so other technical issues. Moreover, Parkinson's disease is characterized by a variety of heterogeneous motor symptoms, and the local field potential recordings from the SDN are characterized by a multitude of features as well that can be related to such clinical symptoms. So the most evident example of is the link between the SDN beta activity and the bradykinesia or rigidity. But there are other evidences regarding the high frequency oscillation, for instance, which might be associated to tremor, or as well as the low frequency rhythms, which might be linked to the dyskinesia. So based on this consideration, what we are now trying to do is to carry out a project whose goal is to link the pathophysiological symptoms of Parkinson's disease to different brain rhythms, with the aim to develop novel spatial informations, with the second goal to inform future adaptive DBS paradigm. So for this project, we recorded subthalamic intraoperative local field potential in 130 hemisphere from 70 awake and at rest patients using these directional DBS electrode. And this is a quite, let's say, large cohort for studies in humans. And we then associated the anatomical coordinates in a normalized space to each of the contact of the electrodes. And we then performed a spatial characterization on the multiple frequency bands, which includes the hotspot localization, the power gradient and focality. And at the end, such spatial metrics is used to predict what is the clinical response to the DBS. So we also give a clinical evaluation of how it works. Sorry for that. So first of all, we extracted the anatomical coordinates of the contacts, which were showing the highest power in different frequency bands. And we evaluated the amount of focality of this cluster of coordinates. And we have found that the focality based on the Euclidean distance between the three axes being higher for HA4, as you can see here, fast gamma and delta band. While for low beta, this light blue here, this was the band with the lowest level of focality, thus meaning a more wide distribution of the activity within the SDN. And going more in details, we found frequency bands from delta to low gamma being more focal in the medial lateral axis, with the exception, again, of low beta, while fast gamma and HA4 were more focal in the posterior anterior axis and in the inferior superior one, respectively. So we also measured the gradient of the power within the normalized SDN space for all of the frequency bands. And we have found a negative gradient for all of the regions that have been measured in the medial lateral axis, as you can see here, which was significant for both HA4 and fast gamma. And we also found, very interestingly, an opposite level of gradient comparing the beta band and the highest frequency band in both the posterior and anterior and in the inferior superior axis. These might have a clinical relevance, since it is known that power in the beta and the HA4 bands have opposite correlation with the clinical efficacy of DBS. So the main goal of our study is also to develop a spectral topographic map of the SDN. So to do so, we separated the contacts accordingly with the amount of their relative power in each of the frequency bands which we considered. And we computed a probability density function for each of the spatial coordinates accordingly with their relative power. So finally, the peak of this distribution in each axis for each band is what we use then to call hotspot. And the result of this process is a spatial representation of the local field potential spectral feature in the three main axes, X, Y, and Z. So what we have found at the end is a distribution of the different spectral hotspots within the SDN. And this, as you can see here, was mostly pronounced in the superior and inferior axis, where the beta frequency was more located superiorly than a random surrogate distribution, while the fast gamma and HA4 were found to be more inferior compared to them. So then we compute the hotspot regarding the clinical outcome of the stimulation, using the same exact methodology used for the spectral hotspots. But in this case, we use the contacts showing the highest clinical efficacy calculated for the rigidity in order to estimate the probability density function. And here you can appreciate how the contacts showing the highest clinical efficacy were very well separated in space compared to the ones exhibited in the lowest level, so these yellow dots here. So we then of course, evaluated the amount of SDN space shared between the spectral hotspot and the clinical hotspot. And we thus found that the position showing the highest power in the beta band, so the beta hotspot, were also the one more close to the clinical hotspot, while an opposite result was found for both fast gamma and HFO, as expected. So we finally ranked the spectral feature based on their relationship with the clinical efficacy, and we use a cross-validation approach, and we then developed an elastic net regression in order to quantify what was the ability of the distance to each hotspot to predict the clinical efficacy of the DBS, so giving a clinical value to all of our somatotopic map. And we found the distance to low beta to be negatively related to the clinical efficacy. So as close as the contact is in the position with the highest low beta, as higher the clinical efficacy is. And the opposite was found for fast gamma and HFO, for instance. So we then evaluated how good was our model to estimate the highest clinical efficacy based on the distance to the hotspots. And this was done in a naive dataset, for instance. And we found that taking the best feature up to the fourth ranked one still guaranteed the highest level of the clinical efficacy for those electrodes. And this was true also when combining different features up the combination of the first and the second one together. So yeah, by concluding, we at the end provided evidence of a spectral topographic map within the SDN, and we found that different spectral features had their own focality and gradient metrics, and also the spectral somatotopy might be used to inform DBS programming because it's directly related to the clinical efficacy of the stimulation. And some how to look, and the clinical and the spectral profiles of the patient are very heterogeneous. So we think that the next generation DBS is really personalized medicine. So then our spectrospatial characterization of the biomarkers is critical to implement and refine the brain sense-based deep brain stimulation protocols. So I would like to thank my Bernese network here at Inselspital, as well as my group. And I'm open for every question that you have.
All right, thanks a lot for the talk. It was very interesting. So once again, I invite everyone in the audience to ask any question that you might have directly in the chat. We still don't have any question at the moment. I have a short question. There is something I didn't really get right at the beginning of the presentation when you talked about the complexity of DBS programming, and you mentioned some ring programming. I didn't really get what that was about. Yes.
So I'm not a clinician, but I can explain you briefly. So basically what they do is to test all of the combination of contacts, and to test the combination just by stimulating all of the possible combination, right? And they see what is the effect of the stimulation on all of the possible combination. And the problem is that the most used technology of electrode has just 4 electrodes, so just 3 combination maximum of to be tested. And so at the end, when the clinician use this technology in order to do the DBS programming, they prefer to use all of the 3 electrodes that they have at the one level. So you can imagine this electrode divided in 4 levels, and they deliver the stimulation just at the level that they decide without comparing the effectiveness of all of the combination. And so they're not really exploiting the technology in this way. So,
all right, get it. Thanks a lot. We have a question from Gerd.
Gerd.
So maybe you want to ask it yourself, or I can also directly relate to the speaker. So question from Gerd. Thanks, Alberto. What are your thoughts on the physiological meaning of the spectral somatotopy?
Say it again, sorry.
I
lost the connection for a second.
Oh, sorry. what are your thoughts on the physiological meaning of a spectral somatotopy?
Yeah, actually, this is very interesting, and we are still exploring this thing. The fact that we have found the beta hotspot, for instance, being more located superiorly and in the dorsal part of the SDN is not, let's say, it's not really novel, and it might have some neurophysiological evidence, but the most unexpected result is about the let's say the high frequency oscillation, which might be more located inferiorly, and also this part of the SDN is not really targeted by the DBS electrode. So this is something like an unexplored field, and what we have found here is that probably HA4 are like let's say highlighting a more focal, let's say, activity of the neurons being located in the most inferior part of the SDN, while the beta activity might be more spread around the SDN. And this is probably due to the fact that the beta activity is more a pathological rhythm for Parkinson's disease. So it's more likely to pick up this rhythm around, especially when you implant an electrode within the SDN. So giving, by concluding, just for, for how our spectral somatotopy should inform both the DBS targeting, for instance, and as well as the DBS programming, and as well as the adaptive stimulation paradigm. So we know now where to implant the electrode in order to be like most effective when deliver the stimulation without even measuring the power. So that's the thing.
Thanks. So we have another question from Gerd Tinkhauser. So to both speakers, what do you think are the main challenges for the implementation of ADBS in the upcoming years? And also, how do you think that taking the time course of beta into account might impact the somatotopy? So I guess it was a follow-up question on the first one. Will bursts also cluster together? So I guess we can start with the first question. So to both of you, Alberto and Fernando, what do you think are the main challenges for the implementation of ADBS?
You want to go, Alberto, or should I?
Oh, go for it. I'm curious to hear your answer.
Yeah, well, although I'm an engineer by training, I would say the challenge is more on the clinical side. I think it's just going to be difficult to show that ADBS is better. There are many people that say that DBS doesn't make a whole lot of sense, and in my own experiences with patients, I sometimes have to second that. So sometimes it will be good to come up with fancy ways of trying to only stimulate when absolutely necessary. But a lot of times, especially with PD patients, we observe these very clear effects where the effective stimulation takes a few seconds to set in. And in that case, the whole rationale of ADBS is sort of, thrown under the bus a little bit. So I think there's still a lot of work to be done on the basic concept of what ADBS is, when it should be used, and how to clinically measure its putative benefit.
Yeah, and then just for adding something, I mean, it is not really clear now whether you're adding something to the treatment by using this closed loop stimulation paradigm. So the conventional DBS is really working fine. So that's the point. We need to find a way to overcome this, let's say, therapeutic effectiveness of the conventional DBS in order to say, okay, adaptive DBS is really what we need to do. But for now, there are not so many evidences telling you that ADBS is really working better than conventional one, apart from a couple of works with a very small cohort of patients. And so, and then again, you talked about the clinical aspects, the clinical problems related to adaptive DBS. There are also, I think, that more engineering problems related to the artifacts. So we are now facing these very annoying heart rate artifacts in the local field potential recordings in this novel device, which allows you to sense and to stimulate at the same time. And so all of these problems are coming out once we are testing the new device. So it's my feeling that in the future we'll face a lot of new problems related to the device and then to the clinical aspect of this disease. And then to add something to the clinical one, is not, as you told during your talk, it is not really easy to separate between pathological and physiological activity by looking at the signal. And so it's not really so straightforward doing this job. And so that's why, I mean, the stimulation has to be triggered by using a pathological activity, but it's not that easy to find out what is a good trigger for the stimulation. So these are the main problems in my opinion.
Yeah.
And then I don't remember what was the second question, sorry.
Perhaps if Gerd is in the call and this is not throwing him under the bus, he could give us an answer to the question he posed.
Right, that would be great.
Yeah, definitely, go for it.
Hi guys, I have no camera, but thanks for the talk. So I really don't have much to add to what both of you have been saying, right? There are a lot of engineering challenges, no? I mean, Alberto, you mentioned it, and there are also the clinical challenges in identifying who are the patients who really can benefit from this technology. So I agree with you, basically. Yes, so will adaptive DBS be something that is useful for every patient, or will it be one additional options for a couple of patients? So we will see over the coming years how well this technology will set and how we implement it. But you know, from an academic point of view, it's really exciting. I mean, look at this, what you both presented. No, so academics is heavily exciting, and there's also there's no question that physiologically it absolutely makes sense having it. But the practical implementation and how much energy, time, and engineering costs I have to invest in this technology to have maybe, you know, how much percentage improvement in a clinical symptom control. So these are really relevant questions. But I think we also need to keep in mind that with this research, with adaptive DBS, we learn how the brain is working. So also it's also this part I think to consider. Yeah. But yes, lots of work. So until our retirement, I think we will all be busy with trying to figure this out.
Right.
Yeah. All right. So Alberto, I think Gerd had a short follow-up questions on your previous answers regarding
Fernando had a question.
Yeah, that was me, sorry.
Oh, sorry. So if you want to ask it directly.
Yeah, I was basically wondering, you guys are the lab that looks at beta time course, not only absolute beta power. How do you reckon introducing bursts into your analysis might change the somatotopy? Might the boundaries between a beta-centric area and a non-beta-centric area might be better defined or worse defined? Or how do you think about bursts in these kinds?
Yeah, that would be very interesting to look more in depth into the beta rhythm, whether we can define these beta bars and what is the relationship between, I mean, the spatial relationship of these beta bars within the STN. So we haven't done anything related yet, but I think that is work to be explored a lot. And what I am expecting is that maybe you can be even, let's say, you could potentially be more focal by looking at the burst and less focal by looking at the overall or baseline activity in the beta band. So that's probably crucial to separate what is pathological to what is physiological. Going back to what we said before, and so yeah, it might be very interesting. Yes.
Yeah, but bursts are it might be a thing that the somatotopy changes depending on whether you're bursting or not. Perhaps when a burst comes, it just recruits cells all throughout the STN, and you suddenly get this flash that isn't localized to specific cells within the STN. Basically, the source circuitry, the whole circuit, oscillating at beta, and then once the burst subsides, the different clusters of cells can start doing their own specialized thing again. Right. Something like that might also be the case.
Yeah, so there might be the trigger for the maladaptive activity that you have in the STN.
Yeah.
Okay.
All right. So I think we don't have any further questions from the audience. So I would like to give a big thank you to Alberto and Fernando for accepting to be our speakers today and for giving two very insightful presentations regarding deep brain stimulation. And we're really looking forward to see what your next results and the next applications will be. So thanks a lot to everyone in the audience for attending today. Thanks again to Alberto and Fernando and to Gerd for the insightful questions. Thanks a lot, and we will see you next month for the next NeuroMeetup talk. Thanks a lot.
Yeah, thank you, everyone.
Thank you.
Thanks a lot. Bye.
Bye, guys.